So as was explored earlier, often the different divisions of the autonomic neurons have contrasting effects. For example, the sympathetic division constricts most blood vessels, but dilates the blood vessels around the heart, the coronary arteries. And both of these effects are achieved with norepinephrine. How is it that norepinephrine can have contrasting effects? Well, that depends on the type of receptors that are found on the target organs. The sympathetic and parasympathetic fibers secrete different neurotransmitters, and the target cells respond to the same neurotransmitter differently depending on the type of receptor that they have for it. All autonomic fibers secrete either acetylcholine or norepinephrine, and there are two classes of receptors for each of these neurotransmitters. Let's look at acetylcholine first. Acetylcholine is secreted by all preganglionic neurons in both divisions and the postganglionic parasympathetic neurons. These are called cholinergic fibers. Any receptor that binds the acetylcholine is called a cholinergic receptor. There are two different types of cholinergic receptors. There are muscarinic receptors and nicotinic receptors. So again, when we look at acetylcholine, we know that it's secreted by all preganglionic neurons in both the sympathetic and parasympathetic divisions. These are the green neurons you see. And acetylcholine is also secreted by the postganglionic neuron in the parasympathetic fibers. Now, how do they have a different effect? Well, that depends on which type of receptor. We'll see muscarinic receptors in things like cardiac muscle, smooth muscle, in glands. And then we'll see that this is either excitatory or inhibitory, dependent on the subclasses of muscarinic receptors. So you can see it gets quite confusing what the response might be. So we have two different acetylcholine types of receptors and then subclasses of those receptors that could either have an excitatory or an inhibitory effect. So how do you know what's going on? Well, at this point, we'll probably just let it go, and you don't need to understand exactly what type of receptor the target has. Now, when we consider nicotinic receptors, they occur at all synapses in the autonomic ganglia where the preganglionic neurons stimulate the postganglionic cells. They're also found on the cells of the adrenal medulla and at neuromuscular junctions for skeletal muscle. Again, you don't need to memorize where each of these are found, but it's just kind of nice to know a little insight in general as to where we might find these. Nicotinic receptors get their name for another botanical toxin that was helpful in their discovery, nicotine. Now, norepinephrine was our other neurotransmitter, and it is secreted by nearly all sympathetic postganglionic neurons. Now, we say nearly all because there are some exceptions. A fiber that secretes norepinephrine is called an adrenergic fiber, because norepinephrine and noradrenaline are the same substance. The receptors for norepinephrine are called adrenergic receptors. We'll see alpha adrenergic receptors and beta adrenergic receptors. Of course, there are subclasses of each of those. There's a table in your text that shows all of the different types and subtypes of these sympathetic and parasympathetic nervous system neurotransmitters. Our alpha adrenergic receptors are usually excitatory receptors, and our beta adrenergic receptors are usually inhibitory. But that's not always the case. Again, it depends on which subclass and where it's found, how it's going to act. Here's an example. Beta adrenergic receptors are usually inhibitory. For example, norepinephrine will relax and dilate the bronchioles, thus enhancing the respiratory airflow. But when it binds to the beta adrenergic receptors of cardiac muscle, it has an excitatory effect. Now, both of these effects may seem to act in contrast, but we're going to increase pulmonary airflow and have a stronger and faster heartbeat, which is obviously appropriate for a state of exercise. So again, it depends where these receptors are, what type of effect they have. So take a moment, close your book, and review here. There are two types of neurotransmitters and two main types of receptors for each neurotransmitter, as well as two subclasses of receptors for each neurotransmitter. So what do you need to take home from this? First of all, which neurons of the autonomic nervous system will secrete acetylcholine? That's all preganglionic fibers and the postganglionic fibers of the parasympathetic system. What are the two types of receptors? Muscarinic and nicotinic, which could be either excitatory or inhibitory depending on which subclass and what tissue they're located in. Now for norepinephrine, which neurons secrete norepinephrine? All sympathetic postganglionic fibers. Occasionally you'll see a postganglionic fiber in the sympathetic system secreting acetylcholine, but let's just go with it being norepinephrine. What two types of receptors are there? There's alpha adrenergic and beta adrenergic receptors. Usually alpha is excitatory and beta is inhibitory. You may have heard of beta blockers. That's a class of drugs that's specific to blocking a particular type of beta adrenergic receptor. Take a moment now and diagram, similar to the picture here on the right, parasympathetic and sympathetic relationships of nerves, whether they secrete norepinephrine or acetylcholine. Knowledge of each of these different receptor types is vital to the field of neuropharmacology. Many naturally occurring drugs bind selectively to one or another class or subtype of receptor. For example, atropine binds only to muscarinic receptors, and curare, a nerve toxin, only to nicotinic receptors. Many synthetic drugs are designed to be similarly selective. The autonomic effects on glandular secretions are often an indirect result of their effect on blood vessels. Many of them begin as a filtrate of the blood, which is then modified by the gland cells, and they increase the blood flow through a gland, such as a salivary or sweat gland, which will increase the secretion, and reducing the blood flow will reduce the secretion. So vasodilation acts to increase blood flow, and vasoconstriction acts to decrease the blood flow. The sympathetic effects tend to last much longer than parasympathetic effects. Acetylcholine is released by the parasympathetics and broken down quickly at the synapse, whereas norepinephrine that's released by the sympathetics is reabsorbed by the nerve, and it diffuses to adjacent tissues, and much passes into the bloodstream. Many substances that are released as neurotransmitters can modulate norepinephrine and acetylcholine function. Although the autonomic fibers will secrete either acetylcholine or norepinephrine, many of them also secrete these neuropeptides that modulate the function of acetylcholine and norepinephrine. For example, the sympathetic fibers will also secrete enkephalins, substance P, neuropeptide Y, somatostatin, neurostatin, or gonadotropin-releasing hormone. You may remember some of the functions of these from last semester, but it's not important at this point. The parasympathetic fibers can stimulate the endothelial cells to release the gas nitric oxide. This gas causes vasodilation by inhibiting smooth muscle tone. This is particularly important in penile erection, and it's the means of the action of Viagra to stimulate nitric oxide secretion, so that the vessels can dilate and blood can pass into the penis. So most of the viscera receive nerve fibers from both the sympathetic and the parasympathetic divisions. And thus, they have dual innervation. This dual innervation can have antagonistic effects or cooperative effects. In antagonistic effects, both systems oppose each other. For example, the sympathetic division will speed up the heart and the parasympathetic division will slow it down. The sympathetic division inhibits digestion and the parasympathetic division stimulates it. The sympathetic division will dilate the pupil and the parasympathetic division will constrict it. In some cases, these effects are exerted through dual innervation of the same exact effector cells. That's what we see in the heart. However, what we see with dilation of the pupil is that each division innervates a different effector cell that has the opposite effect on the organ function. In the case of cooperative effects, the two divisions act on different effectors to produce a unified overall effect. For example, in the production of saliva, the parasympathetics increase the salivary serous cell secretions, and the sympathetics increase the salivary mucus cell secretions, both of which are necessary components of saliva. Even when both divisions innervate a single organ, they don't always innervate it equally or exert equal influence. For example, the parasympathetic division forms an extensive plexus in the wall of the digestive tract and exerts much more influence over it than the sympathetic division does. In the ventricles of the heart, we see a contrasting effect. There's much less parasympathetic than sympathetic innervation. And that makes sense because, as we know, the heart's endogenous rhythm is set around 100 beats per minute. It needs constant slowing down by the parasympathetic division. Dual innervation is not always necessary for the autonomic nervous system to produce opposite effects on an organ. The adrenal medulla, the erector pili muscles, sweat glands, and many other vessels receive only sympathetic fibers. The most significant example of control without dual innervation is the regulation of blood pressure and the routes of blood flow. The sympathetic fibers to a blood vessel have a baseline sympathetic tone, which keeps the vessels in a state of partial constriction or vasomotor tone. An increase in firing frequency causes vasoconstriction, while a decrease in firing frequency causes vasodilation. This allows blood to shift from one organ to another as needed, simply by changing the firing frequency of the sympathetic inputs. The sympathetic division alone can exert an opposite effect on the target organ through control of blood vessels. For example, during stress, the blood vessels to muscles of the heart dilate, and the blood vessels to skin constrict. This figure shows us the example of sympathetic system and vasomotor tone. When the sympathetic division prioritizes blood vessels to skeletal muscles and heart in times of emergency, those vessels will dilate, while blood vessels that supply the skin will constrict. This could be to minimize bleeding in case injury occurs during a fight or flight response. So take a moment here to consider some cooperative and antagonistic effects of the sympathetic and parasympathetic nervous system. Now would be a good time to put all the pieces together for your diagram this week. Remember, we'll be looking at the parasympathetic and sympathetic divisions of the autonomic nervous system. I've given you a few organs to consider. You want to consider how they're innervated. Are they innervated by the parasympathetic, the sympathetic, or both? Are there antagonistic effects or cooperative effects? As well as how long is the preganglionic and postganglionic neuron? What neurotransmitters might it secrete? So you can see we're putting this whole chapter into one diagram. Please remember, make it a diagram and explain. That means a central figure with little blurbs of information surrounding it, rather than simply drawing a picture and writing a paragraph about it. The autonomic nervous system is not an independent nervous system. All of its output originates in the central nervous system, and it receives input from the cerebral cortex, the hypothalamus, the midbrain, pons, and medulla oblongata, as well as the somatic branch of the peripheral nervous system in spinal reflexes. Even if we usually cannot consciously control the autonomic nervous system, it's clear that the mind does influence it. For example, anger, fear, and anxiety. They're powerful emotions, and they influence the autonomic nervous system because of the connections between our limbic system and our hypothalamus. So, knowing this, you can then understand how it's possible, through the cerebral cortex, to modulate other autonomic functions also. The major control center of the visceral motor system, or the autonomic nervous system, is the hypothalamus. It contains many nuclei for primitive functions, things like hunger, thirst, and sex, as well as thermoregulation and emotions. Artificial stimulation of different regions of the hypothalamus can activate a fight-or-flight response, or activation of other centers could have a calming effect that's typical of the parasympathetic. The midbrain pons and medulla oblongata contain many of the nuclei for cardiac and vasomotor control, as well as salivation, swallowing, sweating, bladder control, and pupillary changes. And finally, the spinal cord. It manages autonomic reflexes such as the micturition or urination reflex. Defecation, erection, and ejaculation are all integrated in the spinal cord. We'll explore these further in chapters next semester. Fortunately, the brain is able to inhibit defecation and urination consciously, but when injuries sever the spinal cord from the brain, the autonomic spinal reflexes alone can control elimination of urine and feces. The external sphincters that control urination and defecation are skeletal muscles, so they're under voluntary control. However, the internal sphincters are autonomically modulated. So this brings us to the end of our chapter on autonomic nervous system function.